Hello, my name is Anthony Repay, and I am the current laboratory director here at Method Testing Labs located in Brandon, Florida. I appreciate Laboratories for the opportunity to speak about something that's very passionate to me, which is cannabis microbiology. Some background on myself is not only am I a microbiologist previous to this being in the laboratory director role, I was a food and environmental microbiologist. So taking a lot of what we've learned in the food and the environmental industry and kind of appropriately putting them in the cannabis where it can fit. So involved in ASTM, AOAC, expert review panelist for CASP, and looking forward to kind of sharing some of this microbiology knowledge with you. A quick overview of what we're going to talk about today. First, we're going to go through the fundamental concepts of cannabis microbiology. The second, we're going to go over the traditional culture-based techniques, and then an overview of some of the molecular-based techniques that are on the market currently. As things change in the market, I feel like it's not only a good sense to understand two different types of microbiology. First, a culture-based, what some people will call it media-based, and secondly, the fast appropriating. Melanie, I'm going to go back on that. Just going through an overview of the presentation here. The first thing we're going to talk about are the fundamental concepts of cannabis microbiology. The second is going to be an overview of what we call traditional culture-based techniques. Those are sometimes called media-based microbiology. And the second is where we're going to talk about molecular-based methods of microbiology. In cannabis, we have a broad spectrum of molecular-based approaches that meet the requirements that we need from a state-to-state basis. On this slide, you can kind of see the expansion of cannabis and how it occurred throughout the United States. I think it offers a really cool perspective of understanding that, of course, it's federal illegality of it, but also as states have legalized or allowed decriminalization or allowed for medicinal treatment only, each state had to work independently to allow for its own sort of safety and compliance testing. So I really enjoy this little diagram of the United States. It just kind of shows how we've come, and I think it stops off at 2016, and we're in 2024 now, and it's we're already talking about more medical opportunities and recreational opportunities, adult use. So microbial contaminants differ from state to state. So as stated, due to the federal illegality of cannabis, each state offers a different kind of compliance package, if you would. So currently 20 states test for Aspergillus, 29 states test for STEC, which stands for Shiga toxin producing E. coli, 35 states for Salmonella. These are on a detect, non-detect basis, or a present absence basis. There are 22 states that test for aerobic plate count, 16 states that test for total coliform, 24 states that tell broad spectrum total yeast and mold, and then 3 states for Listeria mono. The aerobic plate count, the total coliform, and the total yeast and mold are threshold-based methods. So they'll be set at some sort of threshold. So in the state of Florida, for instance, the total yeast and mold threshold is 100,000 CFU. So no flower or derivative or edible can be on the market with a higher count that was received in the testing process. So those are kind of threshold versus present absence. So, and we're going to move in real quick to the fundamental concepts of cannabis microbiology. The first one is not only these concepts fundamental to cannabis microbiology, but they're also just microbiology as a whole. So I want to make sure that we take it all the way back and we understand these concepts of where they can kind of take place. First being aseptic technique. So aseptic technique is a method that involves target-specific practices and procedures under suitability-controlled conditions to reduce the contamination from microbes. It is a compulsory laboratory skill to conduct research related to the field of microbiology. What we're doing here is making sure that we are not inducing any sort of high bias or contamination from the human element or the environment around that we subject these samples to while testing. Everything that we use in the microbiology lab should be sterile. And there are several ways that we can kind of go about sterilizing and confirming aseptic technique. So in the past, people would do, if they were reusing things, they would flame sterilize, especially if we're pouring augers or using loops, reusable loops. We would flame sterilize, and we would go about that to confirm sterility. Nowadays, with the broad spectrum, with the broad amount of just one-time use objects that are already sterile, we can autoclave, and also wiping down the benches with IPA, isopropyl alcohol, and a 10% bleach. I always say that you should kind of alternate just to make sure that we're not building up any sort of resistance that can occur. Secondly, because we generally are testing for two different kind of organisms, bacteria versus fungi. What are the difference? And this is, again, just a real brief approach to this here. Bacteria are single-celled microorganisms that lack a nuclear membrane and are metabolically active and divide by binary fission. Of course, just like with anything, there are exclusions to everything, then there's still bacteria, and then the same thing with fungi. Fungi are a group of eukaryotic microorganisms that belong to a kingdom of fungi, obviously. They are categorized by cell type, cell wall, growth form, and reproduction and nutrients. And as we take a kind of a deeper look here and then go into kind of the testing requirements, this is Aspergillus species. Aspergillus, of course, being a mold, is encompassed in a total yeast and mold test. So more often than not, when I say that we're testing for Aspergillus, we're testing for what we call the top 4 or the toxin-producing strains of Aspergillus. So looking at these, this diagram here that I have, we first, in some states, test for total yeast and mold. And then encompassed in that, there are over 100,000 different species of generalized yeast and molds. Okay. In that smaller circle, we have the 180 species of Aspergillus. And then even smaller in that circle, we're testing for what we call the toxin-producing strains of Aspergillus. And those are Flavus, Fumigatus, Niger, and Terreus. Going into bacterial, this is Salmonella and Stech. Again, what I stated before is Stech stands for Shiga toxin producing E. coli strains. And this is where things begin to get a little bit more layered. The first being Enterobacteriaceae is the largest organism here that we're testing for here. That encompasses Salmonella. As we go into the smaller circles, we have total coliform. So that would be anything that is generalized as a coliform. Okay. In the smaller circle, we have Escherichia coli. There are so many different strains of E. coli. And then again, we test for, in cannabis more often than not, Stech, which is the Shiga toxin producing E. coli strains. So you will see that some states do have a threshold test for generic E. coli. Looking at the diagram here, that would be on just those green circles. Okay. Some states have a Enterobacteriaceae test. That would be the whole blue, which also encompasses the yellow, the green, and the orange. And then, of course, total coliform. When looking at this kind of diagram, it kind of goes into the square rectangle argument. So, a square can be a rectangle, or a rectangle can be a square. The kind of thing of like how we can look at enterobacteria and coliforms. So, especially when we're looking at the regulations and how these different microorganisms not only live, but how will they affect on when we're testing these. So, for instance, Nevada, has an Enterobacteriaceae EB and a total coliform test, threshold test. So more often than not, those tests will show similar organisms. We also have matrix interferences that play heavy roles into testing for cannabis. Okay, so I added a nice, really beautiful picture of cannabis flower here. But what we can see here are the trichomes here. And matrix interference, not only on the chemistry side, but on the micro side, can yield not only false negatives, but also false positives. So as microbiologists, using what we know already, we must have ways of combating this matrix interference. The first being a primary dilution. So when I say primary dilution, more often than not, what I'm talking about here is diluting through by 9 mils, by doing serial dilutions 1 to 10, and diluting out the matrix. Now, the important thing we need to make sure we're doing here is we're mixing appropriately, and we're also still meeting the detection limits and also the action limits that the state are giving us. The second may be a secondary dilution. This more often than it takes place in the case of a molecular base where you've extracted the DNA, you have your DNA, and it may take another dilution on there so that you can get that proper hex signal or internal control signal. Always check with your manufacturer from on a both media or culture based, or if you're strictly molecular or vice versa, before just if you're having these matrix interferences. The second thing would be filter bags. So microbiology should be using filter bags where you can weigh on one side of the bag, add your diluent, homogenize with your hand or stomacher, and then you can then take the elutant from the other side to completely avoid taking up a complete cannabis leaf. Again, the trichomes and the contaminants and all of the target organisms will still freely flow through this membrane, but the larger objects are not going to be able to disrupt your pipetting. Also, when doing culture-based or media-based, staggering your open reading frames. So understanding that multiple dilutions are better, so you can compare to see if there is matrix effects that are being carried throughout the dilution, and just being cognizant of those. Next thing is increasing quality control. So here I've kind of subcategorized between media and molecular. So on the bottom, the largest arrow, you'll see the media blank. And then, so that is truly just your sterile diluent. So if you're plating on, your Petri plates and you're using Butterfield's phosphate, or buffered peptone water or something along those nature, you want to make sure that you're also going with 1 mil of your sterile diluent as a control for the media, to ensure that you're not putting any sort of bias on that. You also should use a laboratory controlled spike. Okay, and that's also in the AOAC ALAC Guide to Interpretation for ISO 17025. If you're going abide with the ALAC, is a laboratory controlled spike, whereas your target organism should be also incubated with it. So in Florida, we test for total yeast and molds. We have a certified reference material that comes in a pellet. We incubate it along with on the plate, obviously. So we hydrate the pellet and then we plate it onto the plate. And so what we're doing is we get the samples that come out on the plates. First thing we're going to look at is our QC. Okay, my blank is okay, and put an eye on it. And laboratory controlled spike is within acceptable range. So I have no low bias, high bias when it comes to the method that we put in. So now I have other internal QCs, replicates, and things like that. But at least I know that the start and our process was good. The second thing is a negative and positive control when it comes to molecular. What people often get lost in is that we don't do the blank or the laboratory controlled spike in molecular, which you should still do. If you're testing for salmonella, you should still do a media blank and a salmonella incubated with your samples and carried along with, so that it can be toned as a kind of a prep blank. When it comes to molecular, the negative control and the positive control are more of instrument controls. So generally, more often than not, those will come with either instructions from the manufacturer, or they will come from a little vial as like positive control in the kit. Maybe you have to buy it separately. And so what you're really testing here is to see if the instrument is performing. So if we look at QC from a molecular standpoint, the blank will come back negative for molecular. The laboratory controlled spike, if we're testing for salmonella, so we spike with an appropriate amount of salmonella, that shows positive at the end. So I can conclude that my method and my extraction were viable for target organisms. And then I look at my negative control and my positive control, showing that not only did my method work, but also the instrument is working appropriately. So I could go out in days about increasing quality control, but we have quite a bit more to talk about, so I'll continue on here. We'll move on to here to an overview of traditional culture-based techniques. So traditional culture-based techniques generally work through selective agents, pH, and the nutrient composition that is composed of the media to allow it to be selective or differential. When I say selective, it means that only the target organisms will be grown on that. Of course, there always will be kind of caveats that occur, but for the most part, they're selective for those target organisms. So when we look at this, it's actually my favorite plate, the top left corner here. It's DRBC. That is a selective media that allows for just total yeast and mold. Underneath it, you have XLD. To the top right, let me go to the bottom right first. That is just tryptic soy count, or tryptic soy agar, TSA. It's just a standard plate count. So that agar will grow everything. It corresponds to what we would call an aerobic plate count or a generic plate count. So there's no selective agents, no differential agents in that media that allow for any sort of selective. It grows everything that we want. In the top right corner, we see not only selectivity, but differential. So this is a total coliform test, but when you show the UV light on it, you're also going to be able to differentiate which strains are E. coli. In traditional culture-based techniques, the plate count method of choice is always going to be serial dilution. So that's what I was talking about with your primary dilution, is making sure you're diluting these out serially. So not only are you going to be in the open reading frame of the plate, but also you're still going to be in the detection limit for your state. So as you can kind of see here, this is just a diagram of a 1 to 10 dilutions going through. You can see that 1 to 10 is a TNTC, or too numerous to count. The 1 to 100, we're seeing about 10,000 or 10x less. And as we move down, every plate should have at least 2, so that we can kind of see a good feeling of where we are at with these dilutions, and ultimately if there's any sort of matrix interference that could be occurring. Also on the market, there is most probable number analysis. It's a different kind of approach to a culture-based method of analysis. In food and environmental, we actually conduct these, or I should say we used to conduct these with test tubes, and you still could and abide by FDA and EPA methods of analysis. But through differing of dilutions and the amount of tubes that show positive, you can then indicate what level of contaminant is there. And now MPNs often work better at lower detection limits, but they can be ranging through all of any sort of detection that you need just through dilutions. There are automated methods for MPN in both the cannabis and then the environmental and food industry as well. But instead of having test tubes, they are often pre-filled cards or cells that do these dilution factors for us. And then maybe they're read automatically, or read through manual readers. And then we can then use a MPN chart, and all we do is, okay, so I had 5 of these, and then I had 3 of these, and I had 2 of these, and then we slide across, and then that gives us what detection limit or the most probable number that we think of the target organism in that original sample. So Melanie, I'm just going to take a sip here. So moving on from traditional culture-based or media-based methods, we're now going to move into an overview of molecular-based technologies. So on the market right now, more often than not, we're seeing PCR-based methods of analysis that are occurring. Now, PCR is a broad spectrum term. Now we have endpoint PCR, we have real-time PCR, and I'll try to get into the best of what those differences are and how those relate to microarray and things like that. But when we talk about PCR, The main players are going to be the DNA polymerase, the DNTPs, the buffer, the primer, the probe, and the thermocycler, and thermocycling. And if there's one thing we can take away from this talk is that PCR is the method, okay? And so I try to correct a lot of people that the PCR is a thermocycler. And so in just making sure we're calling things with the instrument a prominent nomenclature. And then one of the cooler things on the market right now, not that PCR is not cool, but is a MALDI-TOF MS. And it's kind of this hybridization of bringing in together chemistry and micro. And we'll talk about that and how I think that would be a really cool opportunity for cannabis to kind of move into, especially when we're moving with more specific target organisms. So that we'll talk about laser ionization, ionization, and then we have the acceleration and separation, and that takes during the time of flight, and of course the detector. So when we talk about PCR, there's really 4 main steps. Okay, and they're kind of outlined right here in these 3 kind of diagrams here. The first is that the primers and the probe. So first, of course, we extract the DNA. So that goes through, an extraction process through each method, a method-specific extraction process. So now we're talking, it's on the instrument and what happens. So the primers and the probe bind to target DNA. Okay, now PCR occurs through a process of heating and cooling. The primers are extended on forward and reverse strands of the DNA. The probe is degraded as a result of polymerization, and the fluorescence signal is generated. Okay, so we can kind of see that fluorescence signal on that bottom, or that most right image, and that's where we see that beautiful logarithmic curve occurring, basically telling us that our target organism has been detected. Okay, that has been through amplification. So now when we talk about like MALDI-TOF, so what is happening is through the time of flight, the ions are then separated, okay, and they're actually read very similar to chemistry. So when in chemistry, we talk about, mass specs or triple quads and things like that. So we're using retention time and abundance here and reference organisms to develop what are these target organisms. Now, I don't think MALDI-TOF MS has made its way through into the cannabis industry that much, but I think it would be really a gain in the industry to see this kind of thing happening. And when going back to kind of PCR, I can go back here. Going back to PCR, that's more real-time PCR is when I was outlining the logarithmic. There's also endpoint PCR, and that can even take place in the case of a microarray, where instead of primers and probes, each cell, or excuse me, with the combination of primers, but each cell with a microarray allows for target organisms to be done kind of in this, in conjunction to multiple target organisms. And then we talked about that MS. But in conclusion, microcontaminants in cannabis products present potential health concerns, underscoring the need for rigorous testing products. So at the end of the day, what microbiologists and what compliance testing laboratories test for are things that could make people sick. In Florida, we are currently in a medicinal-only state. So the fact that my product is going to help someone that may be immunocompromised or may have a differing immune system than someone that of able body, we need to make sure that we are doing everything that we can to have valid and defendable results. The cannabis microbiology industry is and will continue to be a very complex industry. And as the industry continues to grow and evolve, more and more matrices are going to come to the market. So it's a challenge for compliance laboratories to understand these matrices, but ultimately to make sure that we're testing for them properly. It is imperative that labs be familiar with robust methodologies so that they can grow and adapt to this industry. And again, I'd like to thank you all for coming. I'd like to thank LabRoots for putting this on. And if you need anything from me specifically, I have my direct email here. I'd love to talk about cannabis microbiology. If you have any questions, please don't hesitate to reach out. And thank you so much for attending.